From Microbe TV, this is TWiP, This Week in Parasitism, episode 258, recorded on the 2nd of May, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about parasites, the protozoan type, not the human parasites. Lots of human parasites around these days. Joining me today from New York, Daniel Griffin.
Hello, everyone.
The tie is blank, right? There's nothing on it. It's red.
No, you must have bad video quality because it is my bright pinkish.
It's today's Friday, right? STD.
Yeah.
Clap.
It's the clap. It's the clap.
Yep.
It's the clap.
Gonorrhea. The video quality is lower here than on the recording.
Yeah. So the actual recording, everyone will be able to, and maybe even David often zooms in so people can see.
In your rear view mirror, you have that thing. Things are farther away than they appear. Is that, or closer than they appear in the rear view mirror?
Yeah, I think that's like a great scene from Jurassic Park, right?
That was just brilliant that someone thought of that with the T-Rex chasing the car.
Yep.
Also joining us from Glasgow, Scotland, Christina Nowla.
Good evening, all. I'm sorry I gave myself away earlier by laughing.
It's okay. People are allowed to laugh, right?
Well, yeah, it's been a long week, so I feel my laughing is maybe a bit desperate.
Here in the U.S., Christina, it's very difficult to laugh anymore.
I know.
These things are so screwed up.
I've actually just been reading some articles in The Guardian that, not good. So I do feel for you.
The world will suffer as we suffer.
Yep.
There's nobody isolated anymore, right?
No, that's
No
true.
one is safe until everyone is safe, Daniel.
And unfortunately, that applies too much. So
here today is paper day on TWiP.
We've got some pretty exciting, pretty dense papers today, Vincent.
Oh, yes, I did struggle, I have to say. It was pure immunology, wasn't it?
Yeah, it's okay. We'll
some
sort it out.
sentences were not English.
Yeah, we'll sort it out.
But I think you've got a theme here, Vincent. Like we were just a little bit of pre-show banter about one of the recent episodes where it was the rabies and how pre-treatment with antibiotics affects the immune response. So
yes.
Really this whole theme of how the gut microbiome, whether it's bacterial or in this case parasitic, impacts the immune response.
That's right. A lot of different things in the gut. Also fungi and archaea.
Mm-hmm. Viruses. There's a virus in there. Yeah.
Today on TWiP, we discussed a phage of Pseudomonas, which suppresses inflammatory responses to avoid clearance.
Oh, wow.
So Pseudomonas aeruginosa in the respiratory tract, right? It has a phage associated with it. It's a lysogenic phage, so it doesn't kill the bacterium. But the phage, when it comes out of the bacteria, has vesicles stuck to it that contain RNAs that trigger TLR3 and immunosuppress the host. They downregulate CXCL5. Very interesting. How these microorganisms associated with us are, they're really changing things.
Yeah,
I should say they're responsible for the way we are.
And it's all connected as well. It's
really,
all
connected.
Really fascinating.
It's all connected. And that's why you cannot shut down USAID, because everything is connected, folks. And if you thought about it, you'd know. But you don't think about anything.
So shall we get into our paper? Are we starting with
I'm going to talk about the U.S. situation the whole episode. Yeah,
that's why I was trying to shut you down.
Yeah, I know you were. You always do. And I was finished, actually, because I was
going to distract you with science.
I know my limitations.
Yeah. I think it's important to talk about these things.
Well, people like it, yeah. But is that a Clint Eastwood movie, Daniel? I know my limitations.
A man's got to know his limitations. Yeah.
Or maybe Paul Newman. I don't know.
No, that's I think that's Clint. Yeah.
Man's got to know his limitations.
All right, we have a cell paper today, which means it's long and full of data. when I'm reading these papers to prepare, I keep saying, how much more till the discussion? How much?
Yeah.
Oh my gosh.
You can always jump ahead. That's like what I do. Like, should
Yes,
I
right.
die while reading this paper, which could happen.
Well,
during
I
this
say
call.
this paragraph, we don't need to talk about this. This is a gut commensal protozoan determines respiratory disease outcomes by shaping pulmonary immunity. So just by the title, you see the protozoan is in the gut and somehow it's shaping disease in the respiratory tract. So this is very interesting. So we have first author is Kyle Burrows and two last authors, Liam O'Mahony and Arthur Mortha, and they come from University of Toronto, University of British Columbia, University College Cork, McMaster University, University of California, Merced, NIH. So an international collaboration. So what's the story here? All right, so your intestine, we have microbes all over and in us. And this is about the intestinal microbiota, which has quite an effect on things outside the intestine. That's the whole point of this paper. For example, the gut microbiota is a risk factor for development and severity of asthma. Okay, but these studies are all associations. It's hard to get direct evidence in people, right? But we'd like to know how this works. How can a gut microbe influence a pulmonary disease? But what we do know is, and Daniel will like this, group 2 innate lymphoid cells are important in the development of asthma, and they're going to be important in this story as well. They travel from the gut and they accumulate transiently in the lung during helminth infections, and they can affect asthma. These ILC2s are tissue-resident cells. They have the marker called GATA3. They produce IL-5 and IL-13 after they're stimulated, right? They have to be stimulated by something. Mm-hmm. And this includes IL-25, a lot of numbers there. But IL-25, which is made by tuft cells in the intestine, can stimulate these ILC2s, and then they make IL-5 and IL-13. And we think that these cells, eosinophils and goblet cells, expand. These are in the lung, of course, following this activation by tuft cells to promote anti-parasite defense. And what happens is the IL-25 drives expansion of the ILC2s in the gut. They leave the intestine via the lymphatics in the blood. They go to the lung, and they can do their thing there. Okay, so that's for helminth infections and asthma. Now, today we're going to use a mouse model to study this. And in mice, Trichomonas species are protozoan commensals in the mouse gut. And there are some relatives of the Trichomonas species in the mouse that are in humans, and they're Pentatrichomonas hominis and Dientamoeba fragilis. I've never heard of Pentatrichomonas, but I've heard of Dientamoeba from Dixon.
Too.
Pentatrichomonas, that's very interesting. And these show signs of commensalism because people who have them are okay, right? Mm-hmm. They don't get sick. So what happens is in the mouse, the Trichomonas species promote the activation of tuft cells, right? And they activate ILC2s, as we just said, and go on and do other things and help infection. So in this paper, they use a specific commensal, Trichomonas musculus, which they call TMU, because we don't want to say that over and over. So this is a commensal protozoan in the murine gut that tunes pulmonary immunity by regulating the eosinophils in the lung. In part, what you're going to see in this paper, helps shield against dissemination of mycobacteria in the mice. And so they say these protozoa are underappreciated in their roles at doing things in other distal organs.
Can I just jump in and ask a question? I don't know if we have an answer, but how many kind of commensal protozoan parasites or protozoan creatures do we have, really? What do we... I mean, I know a few kind of non-pathogenic protozoan, but I'm not really all that...
I don't know.
Clued on, actually, on that. It would be interesting to know. I know blastocystis is seen as a commensal, isn't it?
We have like that whole section, right, of the parasitic diseases where we go through all the different commensals. And I think part of the challenge with how many, of these different ones is how, there's such diversity within these groups that, like, blasto is a perfect one. Like, is every single blasto a commensal or only certain, types? But yeah, I think, the numbers, it's actually pretty extraordinary, the numbers. I don't think we even look for it as often or appreciate, right? And it's not just the gut. I mean, we have commensal, flagellates in our oral cavity as well.
Daniel, is the nana organism one of those?
Endolimex nana.
Nana is one of the ones that's usually a commensal, right? And I think there was that
Hymenolipis nana is a tapeworm. That's probably not... You mean Endolimex nana?
Endolimex nana, exactly. Thank you.
Yeah.
Okay. So they have these mice that are colonized with TMU, and they're looking for effects beyond the intestine. So they look at multiple other tissues and look for eosinophilia, right? And so they see when you have TMU, they have increased frequencies of eosinophils in the blood, the colon, the bone marrow, 3 weeks. So they have to feed the mice. They can either feed them the protozoan or by gavage, or they can have them eat poop from a mouse that has the protozoan, because mice have no problem doing coprophagy, right?
What a great word.
Yes, it is. And so 3 weeks after you colonize the mice, you see increased numbers of eosinophils. And but the TMU is only in the gut. They find RNA of TMU only in the gut, nowhere else. So the TMU is not going anywhere, which makes it a good story. Then you could say, well, how's it acting at a distance, right? So this paper is all about looking at various cell types and how they're interacting to do this. But this TMU colonization, of course, affects eosinophilopoiesis, the production of eosinophils from the bone marrow, the stem cells that are in the bone marrow, but nothing else. It doesn't increase the production of anything else. And so if you look in the bone marrow of these TMU mice, you get increased numbers of eosinophils, and they bear receptors that drive homing. And so, in fact, particularly CCR3 is one of these chemokine receptors that can mediate lung homing of eosinophils. And so they found only eosinophils increased in these mice, and mainly in the lung.
Is that not peculiar on its own? Because usually with protozoan parasites, we don't really get eosinophils increases. Is that...
Yeah, I think in general,
I always kind of think of helminth and eosinophilia.
And tissue. And
tissue.
Tissue invasive.
And so these eosinophils persist for a while. The TMU doesn't last forever. It eventually is cleared. But there is elevated eosinophils and also elevated IL-5 and IL-13 as well in the colons of these mice. So basically, they find this TMU colonization, you get a systemic increase in eosinophils, which is caused by increased production of eosinophils in the bone marrow, and these home to mucosa, and this is caused by an intestinal protozoan. So somehow it can affect type 2 immune cell distribution in the healthy host. So IL-5 and 13, what are they? They're needed for eosinophil survival, and well, that makes sense that they would go up, right? And they're released by ILC2s, among other cell types. And lung ILC2s produce the high levels of IL-5 and 13 after you colonize the mouse intestine with TMU.
Vincent, do you want to briefly tell our listeners what ILC2s are?
Yeah, I did.
Did you? Sorry, I must have.
I could jump in just to give a little more on that.
But give more.
Okay.
Yes, Daniel.
Yeah, I'll give a little bit more because
Sure.
that was the whole like innate immune cells was an area that I spent, too many years looking into. And this is really an interesting. So when we think of lymphoid cells, right, people are probably familiar with B cells and T cells, and there's an innate type of T cell, right? The gamma delta T cell. There's also an innate type of B cell that would be the B1 cell. But there's also this innate type of lymphoid cell that is a non-B, non-T cell, right? And so initially, people used to think of these as natural killer cells, and they go back. You see these ILC2s all the way back in lamprey and bony fish. So this is a really sort of ancestral population, and the ILC2s, we'll just remember them, type 2 innate lymphoid cells that Vincent, you've been talking about, they have this interaction with eosinophils. So they're actually producing certain cytokines like IL-5 and 13 and 9 and 4, which actually promote the eosinophils.
Right.
So that's it. That's my ILC2.
So they spend a lot of time characterizing these lung ILC2s by surface markers and so forth. But their idea is that these lung ILC2s come from the gut, which and they're stimulated by the T-mu in the mouse gut, and somehow they originate there and then they go to the lungs. That's their working hypothesis here.
I mean, I guess that's kind of the interesting ILC2 thing, is though they may originate in the gut and they tend to be stimulated by certain parasites, but they actually traffic through the lymphatic system to other places. And that.
Now, they do an experiment, which I think is very, very definitive and which I can understand and everyone will. They want to prove that the intestinal ILC2s actually go to the lung, right? So they have mice colonized with T-mu for a couple of weeks, and then they extract their bowel. All right, so I guess they make an incision and pull the bowel out, right, Daniel?
Yeah.
And then they irradiate the bowel with UV light, and that labels 30 to 40% of the gut resident ILC2s. Okay, and then those labeled, they put the gut back in, they sew the mouse up, right, and the mouse is fine. And they can find, because of the UV marking, they can find those ILC2s in the lung 3 days after surgery, right? It's just beautiful that you can label these things. Yeah. And so they traffic between organs. This clearly shows it. Now, another factor in this trafficking is sphingosine, right? What's the general class of molecules that fatty acid? Is it a fatty acid? I think so. Sphingosine. They measure serum sphingosine concentrations in T-mu positive mice or 3 weeks. Sorry, T-mu positive mice, control mice or animals, 3 weeks post-infection with T-mu, and T-murus, different trichomonas, not T-mu, even though it has T-mu in it. And so no increase of sphingosine in the serum of mice that get T-murus, but it goes up after you give mice T-mu 3 weeks later. Now, why is this important? Because it's already known that sphingosine plays a role in migration among organs. So with T-mu, you get if you put that in mice, you get an elevation of sphingosine, and that's going to come into play in this story. So then they want to know, okay, so sphingosine goes up. Do we need sphingosine receptors, right? So they have a sphingosine receptor antagonist, a small molecule that they can give to these mice, and will basically inhibit sphingosine receptor signaling. So presumably the way sphingosine works, it binds to a receptor and signaling happens, and that gives you the downstream effects. And so if you give them this inhibitor of sphingosine receptors, it completely blocks the accumulation of ILC2s in the lungs and mesenteric lymph nodes, but not in the intestine, post-colonization by TMU. So somehow the sphingosine is involved in the trafficking, right? So they do some single-cell RNA sequencing and confirm that these receptors are present in the gut and in the lungs, and they then they next transfer splenic wild-type B and T cells, and either sphingosine receptor sufficient or deficient ILC2s interreg2 null IL2 gamma chain receptor, IL2 receptor gamma chain, which is basically a null of the IL2 receptor, mice. Okay, so these mice are not going to make any T or B cells, right? Or NK cells probably also, right, Daniel? I think. Okay, they colonize the mice with TMU, and they find even in mice reconstituted with sphingosine receptor ILC2s, the eosinophils still accumulate. So then this tells you that the receptor 1 anyway is dispensable. But there are a couple of sphingosine receptors, and there's one number 4. If they antagonize that, then that blocks the trafficking. Okay, so one specific sphingosine receptor is important for these ILC2s to go from the gut to the lung. Sphingosine receptor. I like sphingosine. I've known sphingosine for many years because it's inserted in the capsid of poliovirus.
Okay.
Oh.
It stabilizes the capsid, and the capsid will not uncoat unless you get the sphingosine out, which you do by when the virus attaches to its receptor, the sphingosine pops out.
How cool.
And then the RNA comes out because now the capsid is destabilized. Yeah. It's very cool.
That is really cool.
All right, now another molecule you're going to have to hear about is succinate, succinic acid. And apparently there are tough cells in the intestine that can sense succinate, and some commensal protozoa have been reported to activate type 2 immunity through these succinate sensing tough cells. So this is a metabolite.
Where's it coming from?
It's been described as coming from helminths, so it may be coming from the parasites.
Yeah, it says it's a widely and pleiotropically produced metabolite in the intestine. So
okay, so it could come from anywhere, really.
I mean, it could come from the microbiome. I don't know.
Yeah.
Sorry, I'm just asking questions.
Yeah. So they want maybe it's the microbiome. I don't know, but because they do an experiment where they want to know if TMU and its interactions with bacterial microbiota are required for intestinal type 2 immunity. Okay, so they treat TMU colonized mice with metronidazole, right, which ablates the TMU, and that reduces the lung eosinophilia. And then they colonize the mice with TMU, and that's enough to restore the lung eosinophilia. And then they do this in mice that are either germ-free or have bacteria in their gut. And so if you put TMU into germ-free mice, you don't increase the lung eosinophils, okay? So you need the bacterial microbiome. Whereas the conventional mice, which have a microbiome in their gut, you give them TMU after depleting it, and that restores eosinophils in the lung.
Yeah, I mean, a comment on, like, so here we are, the TMU-mediated interactions with the bacterial microbiota trigger lung eosinophilia via succinate. One of the comments I'll make is they say anti-protozoal, antibiotic metronidazole, but that's also going to target your anaerobe. So it's going to have a little broader than just an anti-flagellate.
Yeah, I think that's why they do the reconstitution in germ-free mice, right? So what is succinate involved in this. Now, that's the next question. So they treat conventional pathogen-free mice, so no gut microbiome, with succinate in the drinking water.
Mm-hmm.
It gives you eosinophils in the lungs, partially, without having TMU in the intestine. All you have to do is give them succinate, and the eosinophils appear in the lung. Not as efficient as TMU, but so the conclusion so far is that this accumulation of ILC2s and eosinophils in the lung needs sphingosine, and it needs succinate, and it needs the microbiome to do all of that. And this is different from enteric helminths, which do it slightly differently. As I mentioned in the beginning, they have an association with asthma, right? And there's ILC2s are involved there, but this is a different axis here. Okay, everybody okay so far?
Yeah, let me be Rich Condit at this point and try to get us where we are. So let me see if I've got this. so we've got this model here where we've got this protozoan parasite. Protozoan, we'll just leave it. We won't even call it parasite. Protozoan hanging out in the intestine. Maybe it's doing good stuff. We don't know how much of a bad player, how much of a parasite it is. And somehow we're thinking that through succinate and the sphingosine,
right.
The tough cells are getting the message. They're picking up the message. Maybe the tough cells are involved here. And then the tough cells and the ILC2s are then the ILC2s are maybe going to traffic to the lungs, and then we're going to get eosinophils.
Yeah, that's the idea. I mean, don't forget, you said succinate in the microbiome. The microbiome is also
so
involved.
the protozoans needs the microbiome.
Yeah, probably because it makes succinate, right?
Yep.
Because you can give mice succinate, and that will increase eosinophils in the lung.
Yep.
Yeah.
Which is very interesting. And you know what's interesting is that you need T cells for the eosinophils to accumulate in the lung. So that's another experiment that they did. So they look at eosinophils and ILC2s in rag null IL-2 receptor gamma chain null mice. So that would be ILC deficient mice, and then ILC sufficient RAG2 knockout mice. So RAG2 would have no T or B cells, right? Mm-hmm. So both ILC deficient RAG2 IL-2 receptor null mice and ILC2 sufficient RAG2 null mice failed to show lung eosinophilia after colonization with TMU.
So you need the tripartite interaction between the ILC2s, the T cells, and the B cells.
That's right. And so T cells mediate the accumulation of eosinophils, and B cells promote both ILC2 and eosinophil accumulation in the lung after TMU colonization.
Do we know what TMU actually does to trigger all of that? Are we coming to that now?
No, we don't know. That's a good question, but somehow it's, I don't know, but tough cells are involved because they start all of this. And so somehow they have to be tickling tough cells, but no, they don't end up figuring it out. We're just going to learn about what happens in the mechanism that we got so far. So you can put T and B cells into these mice that don't have them, and that will not establish eosinophilia unless you have ILC2s there. You have to have ILC2s, right? Even if you have TMU there, if you don't have ILC2s, it doesn't work. So they're essential also for this, right? But in the same vein, ILC2s alone will not give you eosinophilia in the lung. You need T and B cells in the microbiota, right? The only thing that works is if you give intestinal ILC2s, splenic T and B cells into these null mice. That restores eosinophilia into TMU colonized mice. So the tripartite is ILC2 cells, T cells, and B cells. You need all of those for the induction of lung eosinophilia. And of course, you need sphingosine and succinate on top of that.
And TMU.
And TMU, of course. TMU, yeah. Okay, they go to triplet, and they look in these mice intestines, and is it intestine? Quantification and colocalization of T-mu in the intestine. They see They see the T cells, the B cells, and the ILC2s are clustered together in the intestine because they're sharing stuff, right? And they see some situations. I don't know what situations there are. If there's a decrease in the relative distance of these cells, these mice don't have as efficient induction of eosinophils. Okay.
So these cells don't normally hang out together.
Apparently not. Yeah. All right. So then they say, what's the mechanism doing this, which is what you want to know. But I'm not sure we're going to get to what T-mu does, right?
That's okay. We can leave that open.
Yeah, you can't figure out everything at once. Right? So the lung ILC2s express high levels of ICOS. What is ICOS? I forgot what that is. It was shown earlier. Oh, inducible T cell co-stimulator. That is, ICOS is an inducible T cell co-stimulator. I don't know anything about that, but they say that these lung ILC2 cells make higher levels of ICOS than non-ILC2 cells. Okay, so they think the lungs are tuning these ILC2s through this ICOS. And then, of course, there is a ICOS receptor and ICOS ligand, and apparently lung B cells make a lot of the ICOS ligand. And so they're thinking that's one of the things that B cells do is they make this ICOS receptor, and that regulates the ILC2s and eventually eosinophils. So they inject blocking anti-ICOS-L ligand antibodies, and this reverts the T-mu-driven accumulation of ILC2s in the lungs. So clearly ICOS ligand and ICOS are important for this trafficking. They also look at MHC molecules and T cell receptor molecules. And interestingly, mice with lacking T cell receptor B or D, they don't show an elevation in lung eosinophils after T-mu transfer. So somehow the T cell receptor, because we know T cells are involved, so somehow the T cell receptor is recognizing something. They look at this. So CD4 positive T cells, if they put those in mice lacking the T cell receptor B and D, it will recover the phenotype. So CD4 positive T cells are the ones that are important, and they think they're a major source of IL-2, which drives the proliferation of T cells. And these ILC2s in the lungs also make high levels of OX40L, which is an activator of CD4 positive T cells. And so blocking OX40 decreases the lung eosinophils and ILC2s after treatment with T-mu. So again, another player, OXL40, which is involved in this long-distance effect. So basically, CD4 positive T cells mediate the accumulation of lung eosinophils. They do it in an antigen-independent way, and it is driven by OX40 and IL-2, two soluble proteins. Okay. So that's how it works, but it doesn't tell you what the parasite is doing or what the T-mu is doing, right? All right, so now they look in mice at whether this protozoan colonization of the gut and then the effects on the lung could in any way affect allergic airway inflammation. So they make the mice inhale dust mite extract, and they get allergic asthma, and that's got infiltrating hematopoietic cells, eosinophils, IgE, etc. So the conventional mice develop these dust mite driven inflammation, But TMU colonized animals do worse. They have higher pathological scores. And if you do bronchoalveolar lavages from these dust mite treated TMU colonized mice, they have higher numbers of eosinophils. So in this particular disease, the eosinophils are making things worse for the mice. Because if you don't have TMU there, they still get airway disease, but it's much lower, right? And so this protozoan-driven eosinophilia can be a problem. It can promote disease.
Can I ask a question?
Yeah.
What is the normal role of eosinophils in asthma?
I mean, they're part of the disease process. So they're part of the pathology. So normally you wouldn't have a lot of eosinophils in the lung, but the inflammatory part of asthma is characterized by a lot of eosinophils.
Okay, thank you.
So they conclude that this protozoan-driven regulation of lung immunity intensifies allergic airway inflammation through this axis that we've been talking about. So this is all good proof that these IL-2C cells go from the gut to the lung. They increase the numbers of eosinophils, which we've shown is dependent on certain cytokines and T and B cells. What about people? So, they're related organisms to TMU, Pentatrichomonas and Dientamoeba. So nobody has looked for them. In patients with allergic airway inflammation, they haven't looked for these related protozoa, right? Because maybe they're doing a similar thing in humans that TMU is doing, or with mice. So they analyze public data sets of sequences of DNA from patients' sputums, patients who have allergic airway inflammation. So they have 29 patients with severe asthma, and they have non-asthmatic patients with bronchiectasis. What's that, Daniel?
So that's when you've had airway disease for long enough that the airways, the bronchioles, right? So trachea goes into larger airways. They actually get damaged, and you have areas of the lung where instead of them being their normal small caliber, they'll actually be enlarged, and then you'll end up with excess mucus. You'll end up with overgrowth of bacteria in those areas.
Okay. So neither of these patients, bronchiectasis or severe asthma patients, had sequences of a cow trichomonas. So that was a negative control. That's good.
Okay. So good negative control.
Good negative control, yeah.
But they found a higher abundance of these sequences from Dientamoeba and Pentatrichomonas in patients with disease compared to not. They also, but not just those two, but they have other protozoa, Entamoeba, Giardia, Entamoeba dispar, etc. They found in one patient's sputum, but no TMU-related sequences. And so 10% of their severe asthma patients have protozoa-specific sequences in them. But none of them had these two related species to TMU, but they found other things.
So we could maybe just do an experiment now, although it might take years, to use some other protozoan organisms to see if that could be replicated maybe in the mice, to see if it's maybe just a protozoan or whether it's kind of genus or, I don't know, family specific.
So there was a low, I'm sorry, I misspoke. There's a low positivity for these TMU-related humans. But it's very low.
Okay.
And they say it's not really convincing because a lot of patients don't have them. And so they said maybe we should be looking in feces, right? Because that's where the, not sputum, right? Because the protozoans are not supposed to be going to the lungs, right?
Yep, that's true.
But this was a convenience database, so They just looked at that. But I think they'll look at fecal databases. I don't know if they have them, right?
Well, probably something that you could get.
Yeah, I'm pretty sure you could, yeah.
So I guess they've got figure 4F. Is that where we are, where they're actually going to look at the feces? So let's see where they say it is possible that fecal samples could yield a higher detection.
Right.
They look at figure 4F and 4G. So, I mean, they sort of, I guess, have this idea that maybe a subset of asthma patients.
Maybe a subset, yeah.
So
then the last experiment they do is to infect mice with Mycobacterium tuberculosis, right? And it turns out, TB is a big deal. 1.5 million deaths annually. I think that's the biggest killer, right, Daniel?
say that again.
1.5 million deaths annually by TB.
So currently it is the single pathogen responsible for the most deaths in the world. Yeah. So it's 1.5 for tuberculosis, HIV, AIDS, and malaria are kind of the next two of that.
And they say that recently eosinophils have been recognized as an important part of the host defense. Mm-hmm. So, their work shows that eosinophils are recruited to the lungs by these gut resident TMU. So they say, let's see if it makes a difference. Mm-hmm. So they colonize mice with TMU. They get their eosinophilia in the lung. Then they aerosol infect them with Mycobacterium tuberculosis. And they show that if you put anti-IL-5 in, you reduce the eosinophil numbers because IL-5 is important for their generation. But the lung colony-forming units of mycobacteria did not differ between the control and the TMU groups. They say, so TMU does not confer resistance to pulmonary infection by MTB. All right. But what they do find is that it inhibits dissemination of mycobacteria to the spleen. So there's a significant decrease of colony-forming units of MTB in the spleen in mice with TMU compared to mice without TMU. And this impaired dissemination correlates with higher eosinophils in the lungs. And so somehow those eosinophils are preventing dissemination, even though they're not reducing the number of bacteria in the lung, but they prevent them to move later into the spleen, right? So they can deplete these eosinophils by neutralizing IL-5 or by knockout of GATA, which prevents eosinophil development, and you cannot restrain the movement of MTB to the spleen. So this is very interesting that this eosinophilia induced by TMU can prevent MTB moving to the spleen. So those are the data. And so basically what we've shown is that TMU, this protozoan gut commensal in mice, it modulates the gut resident ILC2s through its interactions with bacterial microbiome. These activate the gut ILC2s. They elevate sphingosine levels, and they promote them to go to other tissues via sphingosine receptors. They engraft in the lung these ILC2 cells, and they help with OX40, IL-2 T cells and B cells, and ICOS-L. Those help sustain the accumulation of ILC2s, and that leads to increased lung eosinophils, which is not good for this allergic disease, but it seems to be good for preventing TB dissemination. Yeah.
I mean, I like their graphical abstract because this is cell dense stuff, and their graphical abstract. I don't know if you guys,
it's very nice.
But it's kind of nice because it's sort of so here you've got your TMU, your Trichomonas musculus, and we've sort of seen this paradigm before where it's almost like the parasites are farmers, right? They're modulating the microbiome, they're messing with the microbiome, and getting the microbiome to produce certain things, and here they sort of pull out succinate, and then somehow this is driving gut ILC2 cells, which are then migrating right through the lymphatic system. And then when they get to the lungs, they're going to interact with the T cells using OX40 ligand. They're going to interact with the B cells with ICOS. There's even going to be some direct ILC2 interaction with the eosinophil precursors, right?
Right.
And then you're going to end up with the good and the bad, right? You might be driving asthma in certain contexts, and you might be helping to contain tuberculosis in the lungs in certain other contexts. And the interesting thing, so we always think of tuberculosis, right, as pulmonary tuberculosis. And I think part of that is kind of this Western Europe-centric, Caucasian-centric view of tuberculosis, where true, the White Death in Europe and in the US is pulmonary TB. But in other, say, sort of genetic racial backgrounds, disseminated TB is more significant, right? So,
yeah, sure.
So we remember maybe like the Bronte sisters with the, why do I wear a tie and have it up? Because so many people would have it disseminated into the neck and have the draining lymph nodes. I've seen a lot of cases in Africa, over in Asia, and Nepal, where it's disseminated to the bone, to the heart, to the peritoneum, all over the place.
So my dad grew up in Italy, and he contracted TB, and it ended up in his kidneys.
Yeah. Now, how old was your dad? Like, when was that your dad had TB?
So he left Italy when he was like 25.
Okay.
He spent 25 years there. And so when he was 60, he developed colon cancer. And so then both his kidneys shut down. The ureters, right, the tubes from the kidney to the bladder, they got clogged with TB.
Oh my gosh! Okay,
somehow his TB in the kidneys got reactivated, I guess, by the neoplasia, right? So they had to, in his last months, they had to stick a needle in the part of the kidney where the tube comes out. It's fat, right? There's a name for that fat part. The calyx. So they had to go through his back and stick a needle in and have urine drain out that way. Just one kidney, because apparently the other one wasn't even working. But then the doctors told me he had reactivation tuberculosis, and that clogged the one tube, right? So he was a European. And all those years he lived, he was fine, right?
Yeah, but no, that would be an example. It disseminated from the lung. It was in the kidney. Like, one of the number one causes of adrenal insufficiency throughout the world is tuberculosis in the adrenal glands.
Wow. Vincent, did your dad know he had tuberculosis infection?
I think he probably did. Because, they did those time tests, right? The TB.
Yes. Yeah, I still did those at school, actually.
He worked in a hospital. He was a doctor, so he had to have a time test. And I'm sure he had a lung x-ray, and I don't know if there were any Ghosn complexes or not.
Yeah, he may have had that Ghosn complex.
I just don't know. Yeah. But he was allowed to work. He wasn't considered contagious, but they would have no idea that it was in the kidney, right? Because there's no way.
Until his immune system, is compromised, and then it reactivates.
Right. Wow. I suppose he wouldn't look there, really.
Yeah.
So it's kind of tragic in a way that you get, not only you're dying, and you got to have this other problem, right?
And you're now, like, your urine's coming out of your back into a bag. It's tough.
Yeah. I mean, they tried to put a stent from the bladder up to the kidney, but it just kept clogging. Right. So they did that.
Yeah, unfortunately, I have a number of patients that have these urostomy tubes where, they do that. They're basically putting in and leaving a drain, from the kidney externally into a bag. Yep.
Also as a result of TB or other causes?
Many other things. Usually not TB, at least in the U.S. Yeah.
Okay.
Yeah. But often cancers,
What about therapy? So they mention here that there have been clinical trials with drugs that target IL-5, OX40L, and sphingosine receptor. They've been beneficial in some patients with asthma. And that's part of the whole story that we told here. So they said we should re-examine those in the context of protozoan commensals, because they may be even more beneficial. So And also, they say maybe increasing eosinophils might help with TB, right? Dissemination of TB. So there's some practical outcomes of this.
I think that disruption of the dissemination was temporary, though, wasn't it?
Yeah, it is, because I think the commensals are cleared, right? They don't stay forever. So you have to figure out some way to get around that. Yeah. I think can you do your little paper in
I can do
10 minutes?
Yeah, of course. But first I want to say, Vincent, you should have written that summary for Cell, because everything is so much clearer now than it was.
No, thank you. I'm glad.
When I was reading that paper, I'm thinking, I was scratching my head. I was thinking, I need to look up that molecule and I need to look up this molecule.
Well, the problem is every sentence is full of acronyms. It's very hard to migrate through it, right?
Yeah. It wasn't the most accessible paper, but you've made it really, really much more accessible. So thank you.
Good job, Vincent.
Yes, absolutely.
Thank you very much.
And now on to what? The deeper, the rounder?
I know. So I thought, on our podcast, we kind of mostly focus on human parasites, although today we had a murine one. And particularly we focus on parasites that cause quite significant disease. But every now and then I'm just kind of quite keen to give our listeners, and even just for myself, a glimpse into the weird and wonderful world really that is parasitology outside of our maybe narrow focus, but really extremely important focus. So I want to just introduce you to ectoparasites. But before I do that, I should probably actually tell you what paper I'm very briefly summarizing. So this is a paper that has been published in Scientific Reports, and the first author is Leonardi and the corresponding author is Marquez. And I'm afraid I didn't take a note where they work, but it's an open access paper, so we could just look that up and maybe put it in the show note. And the title is it's quite nice. It's the deeper, the rounder body shape variation in lice parasitizing diving hosts. So we often talk about kind of endoparasites like our helminth and our protozoans that we just look at, but we also have ectoparasites, and this is what this wee paper is about. So, and in particular, seal lice. So lice that essentially live on seals. They're kind of very firmly attached to the seals, and they essentially have co-evolved with their host. So apparently, the seals. The seals weren't always marine mammals; they were land mammals at one point. And at that time, they already had those seal lice, and they entered the water and evolved into marine mammals with those lice. So that all happened together. So I thought that in itself is really quite fascinating. And those seal lice, they feed on skin and mucus of the host and also blood. And they are mainly transmitted from adult to pups during haul-outs, so which is when the pinnipeds or the seals, sea lions, fur seals, walruses, pinnipeds is the kind of group name for those, when they leave the water to rest or to molt, so shed their winter hair, or to breed and have pups. So, and the lice apparently synchronize their reproduction cycle with that of the mammalian host, because I suppose it would be quite challenging for a seal louse to lay eggs without them getting lost in the ocean. That's just a speculation. Maybe they have sticky eggs. I don't know. So what I find quite remarkable about these lice is that they don't get washed off as the seals are diving through the water, and they also don't pop, because you think when a seal dives really deep, the pressures are really high, and you think, well, those lice, they might just pop, which they don't, because seals can actually dive really, really deeply. Some species, for example, the elephant seals, they can dive to over 2,000 meters, which I think is close to 8,000 feet in your currency. Not all of them dive that deep, but they are all actually adapted to long and deep dives.
Yeah, 2 kilometers, so over a mile.
Yeah, it is really deep. And, they use strategies like slowing their heart rate and holding their breath for extended periods. And presume, I suppose the lice, they don't have hearts, but they do need oxygen. So they somehow have to hold their breath too. Anyway, in this article, the authors describe and discuss how some of the adaptations allow sea lice to cope with the amphibious habits of their hosts. And maybe that can help us understand why insects are so rare in the ocean. And actually, I hadn't really ever thought about that, but insects are really exceedingly rare in oceans, and we have millions and millions of insects, and I don't know how many species, many that really live on land and also in freshwater, but really only a very small fraction of them are in marine environments. But those seal lice, they don't only survive, but they also thrive, and they have done so for a long time. So I think that is really fascinating. So the seal lice also are adapted to extreme conditions like their hosts, I suppose because the hosts make them. So they have hypoxia when they dive deep. There's high salt content in the water. It's going to be quite cold, and then the pressures are really also quite high. Some of the adaptations are legs. They're very strong, so they can grasp onto the hair really firmly. They also have these spiracles, these kind of breathing openings that can be closed so that the seawater cannot get in. And also make sure that the air is trapped. So I guess that's the seal lice's way of holding their breath. And in the study, the authors investigated if the body shape of closely related lice species that infect hosts with different preferential diving depth. And they did a morphological analysis of 130 individual lice. And what they did, they collected them and then they photographed them with their dorsal side down, so tummy side down, and they did a lot of measurements and then evaluated the data statistically. And so just to give you a couple of results, I'm not going to give you much, but the lice actually varied significantly between the host species. For example, the lice of elephant seals were very large compared to other species, and they think that's in agreement with a general rule of parasitic lice, that the larger the host, the larger the lice. And actually, that's also known as the Harrison's rule, and that applies to lots of other parasitic species too. So another interesting little factlet that might come in handy for your pub quiz at some point. And they actually also really revealed significant morphological differences between lice associated with regular and deep diving hosts, where lice from deep diving hosts tended to be rounder. And maybe that's an adaptation to withstand the high pressures found in the deep ocean, because the rounded shape maybe optimizes the lice's ability to withstand external pressure by redistributing it over a larger kind of plane, and that kind of minimizes the internal energy required to support the body formation. So in a way, the shape makes them more stable. And I guess some of our submersible vessels that dive really, really deep, they're actually all round. So I think that makes perfect sense. So maybe Jacques Cousteau actually already knew about those lice. I don't know. Actually, also, I thought was quite interesting that many other deep sea species have not evolved a round shape. So they can be, they can be normally fish shaped, for example, and they can live at really, really deep depth. So, and the authors speculate that this could be because sea lice have to endure really quickly changing pressures when those seals, they're fast, they kind of go up and down really, quite high speed. So they think that maybe that rounded shape is a result of them having to be really adaptable, whereas a fish that has a fish shape at 8,000 meters, it's always going to be there. So it can maybe evolve into different shapes. So, I don't know. I mean, I just thought it was interesting. I've been thinking, what do I want my take-home message be? And I actually haven't got an answer. I just thought it was just weird and wonderful, really. And I just think, it just shows you how curiosity can drive research and how important that is, because we may not have an application that directly results from that knowledge, but maybe helps us explain some aspects of parasitism of insects in deep waters, for example. I don't know. I just thought it was weird and wonderful. So no particular reason, but I just liked it. So
the take home is deeper and rounder.
Deeper and rounder. That's right.
The deeper the rounder.
Yes, I guess so. So yeah, that's a very short insight into a really interesting.
In fact, that's a good
paper.
Title. The deeper, the rounder.
The deeper the rounder. Yeah.
I also
confirmation of Harrison's rule: the bigger the host, the bigger the parasite.
Yeah. I thought that was really quite nice.
Yeah.
Oh, that explains Orange
Harrison's
Man.
rule.
That explains Orange Man, huh? I didn't say any names.
Big countries breed big parasites.
And we are where we were at the beginning.
It's very good. That's very good. Florida, the state where old people in science goes to die, Daniel.
Oh, I love that. Florida. Yep.
All right. That is TWiP number 258. You can find show notes at microbe.tv/TWiP. The clinical episode is out there, so you should be working on your answers because that'll be revealed soon. I think in 2 weeks, right, Daniel?
Yep, 2 weeks.
And you can send those to TWiP at microbe.tv. And if you like these programs, we'd love to have your support. Go to microbe.tv/contribute. Daniel Griffin is at Columbia University Irving Medical Center. He's also at parasiteswithoutborders.com. Thank you, Daniel.
Oh, thank you. And everyone be safe.
Christina Nell is at Glasgow University. Thank you, Christina.
Well, thank you, Vincent, for making a really challenging paper much more accessible for me. So I'm grateful for that. And I had a good time.
You're on Blue Sky, right?
I am on Blue Sky.
Christina-naula.bsky.social. Check her out.
Yes, do. Sometimes I post every episode, that's for sure.
Very good. Well, you can find me at microbe.tv or on Blue Sky, Microbe TV, one word without a dot, microbetv.bsky.social. We're there. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon.
Another TWiP is parasitic.
Oh, yeah. Dixon would sometimes say, oh, yeah. You remember?